I am Dr. Meena Sethi. In this video, I will explain what is stelar system and its evolution. In this video, we will discuss in detail what is stele, what are different types of steles like protostele, siphonostele, then different categories of protosteles, different types of siphonostele, and how a protostele has been evolved into various types. This we'll discuss in detail. Now, first, we must know what is stele. Stele is the central core of axis, which includes primary vascular tissue, that is xylem and phloem, with or without pith. Now, if we see, this is the vascular tissue. Red one is xylem and yellow one is phloem. Now, this supply is going to leaf and other parts of the plant. And if we cut a transverse section, this will come rounded in outline. And we can explain different orientation of xylem and phloem. So here we can see that this part is the outermost part, epidermis, and this part is the stelar part, stele part. We can see here this stele is delimited from the cortex by a layer called as endodermis. Then inner to endodermis is this layer is pericycle. Then this yellow one, yellowish orangish, is phloem tissue. And in the center red one is xylem tissue. So this orientation of xylem and phloem surrounded by endodermis and pericycle is called stele. And this type of stele is also called as protostele. But if pith appears in the center of this, if pith appears here, like we can see here, pith is there. Pith is the mass of parenchymatous tissue which is present in the center in the vascular tissue. Then this stele is called as siphonostele. If there is no pith, this is called as protostele. But if there is pith, then it is called as siphonostele. This has again same structure, endodermis, pericycle. This is phloem and this is the xylem. Phloem is facing toward the outer side and in the center is present pith tissue. So without pith it is called as protostele and with pith it is called as siphonostele. So stele is the central course of axis which include primary vascular tissue, xylem and phloem, which may have pith or which may be without the pith. Now, to understand the evolutionary process of a stelar system, we must know what are leaf caps. Like this is the stem vascular tissue and this is giving supply to the leaves which are arising from the stem. Now, when vascular tissue is going to the leaf here, and this is diverted like this. So, this area, this is called as where supply is going. So, this will form leaf cap. Supply is going to the leaf. So, stele, if it is growing like this, so this supply is going to the leaf. So, this is leaf cap, and leaf cap I have explained in a separate video and I'll share the link in the description box where you will come to know in detail about leaf caps. Now, we'll discuss stelar system and its evolution. So, we have discussed this stele which is having in the center xylem and phloem surrounded by endodermis and pericycle. This is called as protostele. Now, if this xylem becomes star shape, if it has radiating arms like this becomes star shape like this. Then this is called as actinostele. So, this is xylem surrounded by phloem, endodermis and pericycle. Now, this actinostele further we can see here, this is being breaking up into many patches of xylem into the phloem tissue. Still it is actinostele because this is carrying the shape of star, and when it has been broken up into pieces like this, right? So this is coming in the form of patches like this. Then it is called as plactostele. So this is xylem, red color I have shown xylem, and orange is phloem, then endodermis and pericycle. So this is plactostele. When it has been broken up into pieces like this, right? Then this is called as mixed protostele. So we can see these are the xylem pieces which are embedded in the phloem tissue. So this is called as mixed protostele. So protostele, if it is having xylem in the star shape, actinostele. Now this is star shape, but this is breaking at certain places, again actinostele. Then patches are produced, this is plactostele. And if it is produced into fragments, right, then it is called as mixed protostele. So this is the types of, we can say, protostele. Now we'll discuss about when pith appears in the center of this protostele, then it is called as siphonostele. Now this pith can be made up of parenchymatous tissue. This can be made up of sclerenchymatous tissue. So if from this supply is going toward the leaves, supply is going toward the leaf. So here this part of the vesicular tissue will get separated like this. So leaf cap will be created. So we can see here leaf cap. Then it is called as Amphiphloic siphonostele. So this is Amphiphloic. So this is the supply going to the leaf. So leaf cap is created. So this is called as Amphiphloic siphonostele. So this part is xylem, this is phloem, this is pericycle, and this is dotted I have shown with blue. This is endodermis. So this is Amphiphloic siphonostele. If there are many leaf caps, but they're scattered, not overlapping, but they're scattered, the supply is going toward this leaf, then supply is going toward this leaf, supply is going toward this leaf. So here this vascular tissue will be dissected at certain places. Then we can see here supply is going to this, here, 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 and here. So this has been dissected. This part is xylem, this part is phloem, this is endodermis. Then it is called as eustele. This has been dissected by the formation of many leaf caps. They are scattered but not overlapping. Then it is called as eustele. And here we can see this will be occupied by parenchymatous tissue, which are forming the medullary rays. Now next different type of, we can discuss siphonosteles. Now here we can see this is different from this. Here this is the pith part, this is xylem, and this is phloem, then pericycle and endodermis. And here we can see this part is pith, green one is pith. Now here this is inner endodermis, then inner pericycle. This is outer endodermis and this is outer pericycle, and this is outer phloem, this is inner phloem, and in the center is xylem. So xylem is surrounded on both the sides by phloem. From the outer side this is outer phloem, and from the inner side this is called as inner phloem. So this is called as Amphiphloic siphonostele. So this category is Amphiphloic siphonostele. Now if supply is going toward the leaf, so here cap will be created. Then we can see the next diagram. Now this is phyllosyphonic siphonostele. This is phyllosyphonic siphonostele, and this supply is going toward the leaf. So a gap is created over here. So we can see this is the endodermis, blue color is endodermis. So this is the xylem arc, which is surrounded on both the sides by the phloem. Yellow one is phloem tissue, right? So this is phyllosyphonic siphonostele, when only one leaf cap is there. But if there are many leaf caps, but they are scattered, not overlapping, they are scattered. Now this will be dissected at certain places like this. So small pieces of steles are produced. So we can see this is one piece, this is another piece, and this is also called as meristele. This is called as meristeles. So these are the leaf caps which are present over here like this. So this part is xylem surrounded by phloem, then blue colored endodermis and pericycle. So meristeles they are produced. So this is called as, when leaf caps are there but they are scattered, this is called as solenostele. If there are many leaf caps but they are overlapping also, when they are overlapping, again meristeles will be produced. These are meristeles. But they are overlapping leaf caps. So two rings of steles we can found, irregular. So each meristele, if you see the structure of each meristele, now this part is xylem, surrounded on all the sides by phloem. Yellow color is phloem and this blue color is endodermis, and this one is gap between phloem and endodermis is pericycle. So these are meristeles which are being produced, and they are arranged in more than here they are arranged in two rings. So this is called as dictyostele. Now this stele, which is called as eustele, this is highly advanced and this is found in angiosperms. Rest of the seal, they are found in gymnosperms and pteridophytes. They are also found in angiosperm, but this is highly evolved. And if they, like we have shown, this is dictyostele when meristeles are being produced. Meristeles are small steles, dissected steles. If there are more than two rings, then this will be called as polycyclic dictyostele. So we have seen, if there is no pith, this is protostele, and if there is pith, this is siphonostele, and if this is dissected, this is called as eustele. And this type of stele is found in highly evolved vascular plant, that is flowering plant angiosperm. So this is how evolution of the stelar system has taken place. Thank you for watching it. If you have any doubts or question, you can ask me in the comment box. If you like my video, please like, share and subscribe.